let's begin. Good afternoon, everyone. It is a great pleasure to welcome you to this talk about our BSc degree in Infection and Immunity. I'm Richard Milne. You can see a picture of me on the screen there, sitting in one of the labs upstairs. I'm joined today by two of our current final year BSc students, Mary and Gemma, who you may also be able to see. Do you want to say hello?
Hi.
Hi.
Hello, thank you for doing this. You'll hear more from Mary and Gemma in a little while, but just to kick us off, I just thought it'd be nice to show you where we are. So we are in London, which is marked here on the map, and this picture is the main or the centre of the UCL campus in central London in Bloomsbury. I'm actually sitting in this building right here. This is the Cruciform building, which is where some of our virology labs are. And this sort of dome with the portico, that is the main UCL building in London. And for those of you that know this area, Gower Street runs along here. And our big associated teaching hospital, University College Hospital, is this green and white building right there. So this is where we are. This is where Mary and Gemma are doing a lot of studying at the moment this year, and they'll tell you more about that in a moment. So this is the plan for today. I'm going to kick it off, and my talk is going to have two parts to it. I'll start by talking about some science. And I think that's really important because in the end, that is why we're here. We are here to do really good science and to train students to do really good science. So if you are considering studying this amazing discipline of infection and immunity, listen carefully to the first sort of 10 minutes or so, and that can help you work out if this is the right thing for you. If you're thinking that what I'm talking about is really interesting and you'd like to know more, then this probably is a good choice. If on the other hand, you're thinking, oh my God, how much longer is this going to go on? Then I&I is not perhaps the best choice for you, and you might be better to think about a broader bioscience program, which still gives you the opportunity to choose a specialist direction, but doesn't make that early commitment. So then I'll change tack. I'll briefly summarize the structure of our degree program and tell you a little bit about what we cover. And then I'll shut up and I'll hand over to Mary and Gemma, who are going to do a sort of double act, tell you a bit about their experience, and I think in particular tell you about the research projects that they're doing right now, which are a really central part of the whole program. And then we'll finish in plenty of time for you to ask questions. There should be on the Zoom control panel a Q&A button. On mine, actually, you have to click more and then Q&A comes up. If you could ask your questions using that function, then I will be able to see them and read them out and we'll answer them. And I'd encourage you to feel free to ask questions very broadly. So ask them about the science, about virology, about the program, about university, whatever. Take advantage of this opportunity to get good information. Okay, so I'm a professor of virology. As I said, in this building, we have virologists. They're all upstairs at the moment. And I want to, for the science part, I want to give you a little tiny excerpt from a much bigger story that was developed in this building at the height of the SARS-CoV-2 pandemic. But to introduce this, we need to think briefly about what a virus is and what it has to do. And so to do that, I want to show you this picture. So this is a very basic depiction of a virus, and it illustrates the two main parts. All viruses have some kind of nucleic acid, DNA or RNA, that makes up their genome. and all viruses contain that in some sort of protein container, which we usually call a capsid. There are many, many flavors of this, and we don't need to go into the details today, but that's basically the message. But viruses are interesting because all the biology that you have considered in your studies so far, I guess, has been based on these things, cells. And you know all this stuff. You know that all cells are wrapped up in membranes. They have cytoplasm, mitochondria, nucleus, and so on and so forth. When you, excuse me, when you get into viruses, in a sense, you enter a different biological world because viruses are not cells. They're very, very different. On their own, they really are inert. They don't do any of the things that you have probably learned in your biology lessons that represent or comprise life. But viruses are biological agents and they can replicate. But in order to do that, they need cells. And of course, what they do, as you probably know, is they go into cells and they completely repurpose the biology of the cell from doing its cell thing, which you'll be familiar with, to making new viruses. And that's a pretty cool thing to study. If you want to understand how cells work, quite a good way to do it is to put a virus on and work out what the virus does. Because in a crude and perhaps slightly flippant sense, viruses have been studying cell biology a lot longer than humans have, and they're a lot better at it. And so viruses, in order to replicate, have to go through a series of steps. They have to get into the cell, and that's pretty remarkable. They have to recognize the right cell, get in, and in doing so, not destroy the integrity of the cell. And then they have to express their genes. They have to replicate their genome, so make more copies of their nucleic acid. They have to build new virus particles, and then those virus particles have to get out of the cell, and then they're ready to transmit either to the next cell or to the next host through the common transmission routes that you'll be familiar with. And coughing and sneezing is the respiratory route, is the material one for SARS-CoV-2 that we're going to talk about in a minute. So this is a sort of kind of a depiction of the replication cycle of HIV, which is the virus that causes AIDS. But all viruses do something like this. And if you accept, as I'm sure you can, that for a cell, and indeed for a whole human, being infected with a virus is not a good thing, then you will not be surprised to hear or to learn that cells have evolved to be quite resistant to being infected by viruses. And the way this evolution has happened is that cells have evolved a whole family of proteins and functions that we call sensors. And their role is simply to detect that the cell is infected. And there are sensors that work at almost every step of the infectious pathway that I just described. And I've illustrated those with this sort of yellow shape. I don't know what shape that is. And when those sensors are triggered by detecting bits of a virus or the signals that viruses produce, they respond. And they respond, one way they respond at least, is by triggering the cell to produce a protein called interferon. And interferon is a really, really, really important protein in virology. We call it a cytokine. It's a protein that's produced by cells and released. And what interferon does is it makes cells resistant to virus infection. So you can see why cells which have evolved to try and not be infected with viruses would produce something like interferon. And the way interferon works, and this is really important for the experiment that I'm going to show you, is like this. So here's a cell, a set of cells. Maybe imagine this is in your respiratory tract or your nasopharynx. Here's the cell that I just showed you, but imagine this is in fact a respiratory virus. So this cell gets infected. That's probably quite bad news for this cell. It'll probably end up dying, but it senses that infection and produces interferon, and interferon makes the cell antiviral. And it does that by actually inducing the cell to make hundreds of proteins whose sole function is to inhibit the virus from doing its thing. But interferon has another amazing property that's really important, and that is that it doesn't just work in the cell that's infected. It's actually released from that cell, and it can float around and trigger adjacent cells to do the same thing. They can start producing all these antiviral proteins, but crucially, they're doing that before they've even been infected. And so what happens is that this infected cell becomes surrounded by a kind of barrier of cells that are already largely resistant to virus infection. And that, if it works, arrests the spread of the infection. And interestingly, interferon also works at some level system-wide. It goes into your blood. And if you've ever had a nasty sort of virus infection, flu is the classic one, you might remember having that dreadful feeling of going to bed and just feeling absolutely awful and so exhausted that you can't even lift your arm up to have a drink of water. And that sort of almost feeling so bad that you can't see a way through it. That is actually interferon driving this antiviral response. So next time that happens to you, you'll be able to reflect on the underlying biology and recognize that actually that's a good thing, even though it's quite an unpleasant thing to experience. So I've said that viruses are dependent on cells in order to make more viruses. And I've explained to you just now that cells and organisms, but we're talking about cells at the moment, have evolved to counteract the challenge that viruses present to them. And so you get this extraordinary antagonism where the cell is doing its best to stop the virus replicating, but the virus, if it's going to be able to continue to exist, has to be able to replicate in the cell. And so viruses, all viruses, certainly all viruses that infect mammals, have evolved to be able to suppress or evade the antiviral state that this amazing protein interferon produces. And so you get this back and forth within individuals and actually across all life through evolutionary time, where all of the viruses and all of the hosts are engaged in a kind of grand evolutionary dance where each is trying to do its thing, which requires it to be a negative effect, or which requires it to have a negative effect on the other one. And so you get this back and forth, which is a great place to be if you want to study evolution and how things like viruses evolve. So with that in mind, I want to show you a small experiment that had a very, very high impact that was done in this building in the SARS-CoV-2 time. And this is a picture, that's Lucy and that is Lorena, and they're both working upstairs in one of our high containment laboratories. And this is actually one of the very first experiments they did. And this isn't really even an experiment. This was just when, right at the start of the pandemic, when we got the first sample of the virus. And the first thing you do when you get a virus is grow it. And so they put it into some cells. These are some cells. The little sort of bluey, purpley circles are the nuclei of cells. And we can stain cells to see if they're infected with a fluorescent green dye. And within 5 hours of putting the virus onto the cells, these are airway cells from humans, you can see a bit of green. And then with 10 hours, the virus has spread. And within 24 hours, it's pretty much taken over the whole culture. So if that's in your respiratory tract, you can see why this virus is going to do some damage and spread quite rapidly. So that's the sort of experiment that you can do with a virus. Now, you may remember, although it may have faded away as a bad memory, that just about a year after the start of the pandemic, remember the first case of SARS-CoV-2 was probably in sort of mid-December in China. We started to be aware of it around Christmas time. So a year later, sort of approaching Christmas 2020, there was a report of a new version of the virus, and it was first detected in Kent, in England, in the county of Kent. And that led to some slightly panicked headlines, as I've shown you here. But there was a sense that this new variant, it was originally called the Kent variant, and then quickly it was renamed the Alpha variant when a standardized terminology was adopted. It was clear that this was spreading really, really quickly. And within, I think, 2 or 3 weeks, it had completely replaced the original SARS-CoV-2 virus in the population of the UK, which is an amazing thing. So Lucy and Lorena were in the lab, and they got that virus on Christmas Eve 2020 in this building, and did exactly the experiment I've just shown you. So infected some cells, and this is one of the key experiments. I'm telling you a very reduced version of this story, just to make a key point and to show you the kind of science that we're doing and how we think about this. So what they did was they took 2 versions of the virus, the original virus that popped up in 2019 and had caused the first wave of the pandemic through the early part of 2020, and the new variant, which we call Alpha, and put that into cells and seen how it grows. And here's the experiment. So this is the time, 24, 48, 72 hours. And this is the number of copies of the viral genome. And that's a measure of how well it's replicating itself. So the higher the number here, the more virus replication there is. And I hope that you can see, if I were to ask you to tell me what this graph showed, I hope that you would say that it shows that the Alpha variant, the new one, is growing faster and somewhat better than the original wave 1 virus. So that was a pretty interesting result, a pretty exciting result. Remember, this is during the lockdown when everybody is at home, except the virologists here and the medics across the road in the hospital. And there's this new virus or new strain of the virus that's taken off and is spreading around the country and seems to be better at transmitting. And also early on, people thought it was also causing more serious disease. So this was a pretty important observation. But the thing you want to do as a scientist is go beyond just observing and describing. You want to start to understand. You want to ask why or how. What is the basis of this difference? So we go back to what I said a few minutes ago about interferon. If you have a new strain of the virus that grows better in cells and grows faster, you might make a hypothesis, and this is how we do science. You might say, well, interferon and the interferon response is one of the most important things that's keeping the virus in check. So we hypothesize that this Alpha variant somehow is better at evading the effects of interferon than the original virus. So that was what Lucy and AK and Lorena did. How do you do that experimentally? Well, an easy way to do it is to go to your experimental infections. They're sitting in culture vessels in an incubator upstairs. When we grow cells, we have special medium growth liquid that provides all the nutrients that they need. And anything that the cell produces, will be released into that liquid. And so you can just take a sample of that liquid from each of these two infected cultures, and you can actually measure how much interferon there is present. And that's quite an easy thing to do. Remember, interferon is this super protein that is triggered in response to infection and then initiates this antiviral state. And so here is the, whoops, oh, I've given the answer away. Here's the results. So these are two separate experiments with the original virus. You always want to do things more than once to show that they're what we call reproducible. And what we're showing here is how much interferon is induced above the level that is the sort of baseline. So this is 40 to 60 fold induction. That's quite a lot. But then look at this. Here is what the new alpha variant does. And here's three separate experiments. And this is way, way, way down. This is really hardly inducing any interferon. So that's going to make a big difference, because that means there are going to be far fewer of these antiviral proteins. And so this virus is going to be able to replicate better. And so what we think this showed, or what we know this showed, is that already the virus, this new virus that's just popped up in humans, is adapting. It's learning how to evade the responses that the new host is putting up against it in a way that enhances its replication, and through that enhances its ability to transmit in the population. And that's why it took over. Now, that picture I've just shown you was part of this paper that was published in Nature. There's Lucy, there's AK, and there's Lorena. They're the people that did the work. And there's the graph I just showed you. So this was just one figure of one bigger panel of data, and there were 5 data panels in this paper. But this was a really important paper because this was one of the first that started to explain what was driving the ability of SARS-CoV-2 to spread so effectively in the population. Now, you might say, well, that's all very good, but that was 4 years ago. But look at this. Since then, we've had a succession of different variants of CoV-2 come up, and that's shown on this graph. We had the alpha that I've just talked about. Then there was the delta. Some places there was beta, sometimes there was gamma, and now we're into the Omicron era. And certainly in the early time, if you look over here, each one of these variants did exactly the same as what alpha did, the thing that I've just described to you. But they did that independently. They've all evolved to be better at evading the host response in order to replicate better and transmit better. And that's such an important concept in virology, in infectious diseases, in biology. Really, nothing makes sense in biology unless you're thinking about it through the prism of evolution. And I think that is a very key message that this work generates. So I want to finish this part by just saying, what's coming next? There are a number of things going on at the moment. You might have heard of the Mpox cases that have just cropped up in the UK, part of a global expansion. You might have heard about bird flu going into cattle in the US and then into pigs, and now there are some human cases. And I want to get you to think about whether we can use the science that we've just talked about, measuring how good a virus is at escaping from the host responses, perhaps in combination with the new technologies in AI, to actually go and sample what's out there in the jungle and in the bat caves where all these exotic viruses come from, and ask, can we predict, can we work out which viruses are the ones that are going to cause the problems? And can we preempt the next pandemic? Can we actually make a vaccine before the virus has taken off? And I think that increasingly this is becoming a viable proposition, and it's work like the work I've just described that feeds into being able to do that. So I think that's a very exciting area of research that you may become more familiar with over the next few years, particularly if you come and study on this program. Right. Now I'm going to change tack and briefly describe what the BSc is all about, and then I'm going to hand over to Gemma and Mary for them to give you a bit of first-hand experience. So what we want to do really is present the science of our disciplines, infection and immunity, as far as we can through the lens of the research scientist, through current thinking, current questions. We want to sort of stimulate your curiosity and your creativity and channel them into the main purpose of a scientific investigator, which is discovery. Our job is to discover stuff. And we want you to learn to think in a way that empowers you to understand that and perhaps actually deliver that. So it's a 3-year degree. The first year is very broad. It's taught with lots of other undergraduate sort of biomedical science programs. It's based almost entirely at our Royal Free Hospital, teaching hospital campus, and it's basically about how the human body works. The modules that you do are here, and you can probably see that they're sort of based around human body systems. So this gives you the kind of basic mechanism of human biology, which of course is quite important if you're going to understand how an immune response works and how an infection works. In the second year of the program, that theme continues. There is still some sort of quite traditional, almost sort of generic bioscience content, but we are increasingly starting to focus on the core disciplines of infection and immunology. And one way we do that is by having a weekly journal club, as we call it, where each week the students go through a paper, a recent scientific paper or a part of a paper, and really start to get to grips with that in a very, very detailed way, because that's super important part of doing the science. And implicit in this are these themes that I think of as going through the program. Cutting edge. Where you want to be in science is right at the state of the art. You want to know what people are doing right now, what's going to be published tomorrow. And we try and get students to think like that. And you need to be a confident and effective communicator. You can be the greatest scientist in the world, but if you can't effectively communicate your findings, then in a very real sense, they're rather limited in the likely impact that they're going to have. And critical analysis, we want you to sort of transition from the rather traditional being told something, memorizing it, and then producing it in an exam, to actually becoming a processor of information, to not just accept everything, but to ask, why do you think that? What's the evidence for that? Is there an alternative explanation? And that's all part of this process of thinking like a scientific investigator. So then I think that the climax of the program is the year 3, the final year, which is what Mary and Gemma are doing right now. And this is where we really get to grips with what is happening in our discipline at the moment. And I say we focus on our research and your research. And what I mean by that is that most of the teaching that we do is anchored in the research that is ongoing in our department. Not all of it. There is a certain amount of lecturing about facts. You have to learn some facts, but we can color all of those and illustrate those with our own activities. And in the first term, so October to December, everyone does the same. They do these 3 modules and they start their research project. And then in the second term, January to March, they choose 2 from this list. And this list really reflects the research that we're doing in the division. So we don't try and cover everything like a textbook would. We teach about what we do. And for me, that is why you come to a research-rich university like UCL. You don't come here for me to teach you about something I boned up on the day before in the textbook. You come here to hear about stuff from people that are actually doing that and are really leading the field. And I think we're able to deliver that quite effectively. So what do you get from this? You definitely will understand how infections work, how the immune system works. You'll definitely have some understanding of how the human body works. But I think more importantly, you'll have a real insight into current thinking. What are the important questions at the moment? And how do we actually go about answering those questions? How do we design experiments? How do we come up with new knowledge? And how do we interpret that? How do we critique that? And how do we communicate our findings in an effective and a compelling way? Where do graduates go? I mean, I think in a very real sense, you can do almost anything. I would argue that the set of skills that I've just outlined, that sort of scientific method, if you like, interpreting complex data sets, communicating them clearly in written and oral form, I think that is a very transferable skill set, and I think you can deploy that in almost any setting, actually. But I think it's fair to say that most of our graduates go on to something related to biomedicine, either further study, master's programs, PhDs, working in a lab, or in what you might call allied careers. So science writing, pharma, and so on and so forth. But there are many opportunities that are open to you. So if you want to know more, you can look at our prospectus. I think we'll put the links in the chat thing. The standard offer is 3 A's in biology, chemistry, and we prefer maths, although it's not an absolute requirement. I would really encourage you to come and visit UCL. You can come at any time. It's an open campus. You can book a tour. They're led by current students. That's a good way of getting a good handle on what's going on here. There will be official open days, probably they're usually sort of in late spring, summer. The dates aren't there yet, but you can register your interest and you'll get an email when the dates are available and you can book a slot. So I'd very much encourage you to do that. You want to get as much information as you can, and coming here and visiting and talking to some current students is a very good way to do that. Right, so I'm going to stop at this point and hand over to Mary and Gemma for a few moments, and they're going to say something, although I don't know what they're going to say. I gave them free rein. So Mary and Gemma, over to you. And I think I'm going to unshare the screen, so we'll be able to see you. Go ahead. And sorry, Gemma, forgive me. Just to remind everyone, at any time, do start typing questions into the Q&A box. Anything that Gemma or Mary say that triggers your interest, type a question. And you're very welcome to ask questions about the science as well, and we'll answer those in due course. Gemma.
Okay, well, one of the things that Richard said at the start of the talk was if you didn't enjoy his sciencey bit, this probably wasn't for you. And I think that's so true, especially since this morning we actually had in the class infectious agents, we spent basically 2 hours just talking and going through that paper. So if you did not like it, this really is just not for you.
Did you like it?
Um, I did, yeah.
You have to say
So,
that, don't you?
yeah. But but one of the other things that I really like about this course is that I was just at brunch with my friends yesterday and we were all talking about our dissertation projects because we're all quite STEMmy. And some of the things that we mentioned all really liking about UCL was the fact that a lot of my friends were saying that when they were citing papers for their like research for their dissertation, they were citing people who are actually in their department. And I thought that was especially true for this department because we actually do like hear hands-on from people who are doing these really cool, exciting research things, and we get to do our own. So I'm working on T-cell sequencing, which I know is a bit more on the immunology side compared to what this chat has been, but I'm basically got to spend a lot of time in the lab extracting samples and preparing them to be sequenced. And then I'm a bit of a computational person, so it's quite a mathy bit at the end of the sampling, but there really is something for everyone. So yeah.
So I was just going to
Great.
speak a little bit about why I chose the course, and then a bit about my project. So I was always interested in the pathogenesis of disease and why some infectious agents circulating amongst animal reservoirs can jump into humans, and why only a few of them can then cause pandemics. And then also how human impact affects the dynamics of these viruses jumping between species and becoming pandemics. So things like climate change, they alter the way different animals migrate, they cause contact between humans and animal reservoirs that can cause these jumps to occur more frequently. I actually did my EPQ project on that when I was in college, so that was useful. And then for my project now, I'm doing more infection than immunology for mine. So I'm working on cytomegalovirus, which is a disease that mainly affects immunocompromised patients as opposed to those with functioning immune systems, just because it affects the immune system's ability to obviously contain the virus and stop it from reactivating. So we've been targeting CD13, which is a receptor on a range of human cells, which is believed to interact with GB, which is a protein on the CMV virus, and can help mediate the virus's entry into the cells, and we think also the spread between cells. And we've been doing this currently with a variety of different inhibitors and also anti-CD13 antibodies. We've been doing loads of assays, and we've been looking into that so far. So, yeah.
Great. And do you enjoy doing that? Do you like being in the
I
lab?
do. I do, yeah. Yeah. I also think that in our first year, because we did all the like medical science kind of background, I think it's important because then you can understand how your science is being translated into people, how it affects people's lives, and I still remember that just because of that background as well, so it keeps it
Right,
important.
right.
Yeah.
Good, good. Thanks. And Gemma, what about are you enjoying actually being in the lab? Do you feel, I mean, I'm curious from both of you. I mean, do you feel like you're kind of chucked in at the deep end, or do you feel that you're kind of competent? How does that happen?
Well, my lab would spent a lot of time like training me. So I spent like I think a day just sitting there pipetting water onto a scale to make sure I was like accurately measuring everything.
That's
And
a key skill.
I've done so many like test things of everything. But also a few of the practicals that we've had are stuff that kind of prepared me, although I do think like it's the hands-on experience that really gets you going.
Okay. And can I ask you also, I mean, it's early days, isn't it? Because we haven't got to term two yet, but what about the sort of the non-lab, the teaching and the modules and that side of things? I mean, how are you finding that? Are you enjoying that? What have you
I think
learned?
I specifically enjoyed the modules the way they're taught this year as opposed to the last 2 years, just because we have different teaching styles. So we obviously have the infectious agents module where we get to have discussions in class, and then we have some more traditional, lecture like ones, and then some of them are in big groups and some of them are in small groups. So you feel a bit more like you can have a discussion in those as well. But obviously, that this year is much more tailored to infection immunity. It gets more per year, so I've enjoyed this year the most so far, I think.
Okay, that's good. It would be awful if it wasn't the case. Right. What else? Is there anything else you guys would like to say? What would be your top tip? I'm putting you on the spot for someone who's thinking of applying, I guess, either for this program or just, university in general. What were you thinking at this point?
I think one of the things that I was thinking was kind of how can things be translated if I don't end up really loving it, which I know is not like maybe the best thought to have, but I was a bit anxious applying to schools and kind of figuring out what I want to do. And one thing that I do find good about this program is that you do learn a lot of translational skills, especially into like other areas of science, but also just in terms of thinking for yourself a bit. So yeah.
Okay. Any thoughts, Mary? Go on.
I don't really I don't really know.
That's
I
all right.
mean,
It's I shouldn't have put you on
yeah,
the spot.
I don't have a straight answer for
Okay.
that.
I've actually got a slide. I'm going to just show you this because I thought that this might come up in questions, but I can preempt it. And that is the issue of a specialist degree. I mean, this is a pretty focused degree, certainly by the time you get to year 3. And I would make the following points about this. The first is that although it is very specialist and we have a focus, actually the core content that is delivered, particularly in the first year, is very much comparable to other biomedical sciences, biosciences. You know, choose your biosciences degree, and if you think about it, it has to be, obviously, because you can't go into a specialist topic if you don't have the grounding. So it's not as specialist in a sort of exclusionary way as you perhaps might think. I've made the point about scientific method being a really valuable transferable skill set, but I'd argue actually that in fact our discipline is the antithesis of niche and focus, because I think that our discipline provides almost a unifying framework for understanding all disease. I would argue that all disease ultimately is down to some aspect of inflammation, which is an immune response. So this is pretty universal. And I think infection and immunity is a very interdisciplinary topic. And that's a hallmark of good science. You have to be a cell biologist if you want to be a virologist. You kind of have to be an evolutionary biologist. You have to be an immunologist. It sort of invites cross-disciplinary thinking, and that is a very rich source of creativity and new scientific ideas. So I think, in fact, perhaps paradoxically, we end up giving you a broader view than you might get from a more traditional program. And I guess the last point I'd make, without sort of wanting to sort of protest too much, is that I would argue that the whole point of coming to a university like this is specifically to be taught by specialists about the thing that they're doing. That's really the distinguishing feature of a research intense university, and I think that's that is the kind of thrilling opportunity that a degree like this offers. So I think that's all I'm going to say. Now, I would invite you to type your question. So far, we have one question. So it says, from an anonymous person, how much microbiology is covered in this BSc, and what is the main focus on? Is it mainly on virology or a more general pathogen study? I guess what you mean when you say microbiology is to distinguish that from virology. We are, I mean, I think I made this point. We are not trying to sort of recapitulate a textbook. You know, if you look at a typical microbiology textbook, it'll have 10 chapters on different kinds of bacteria, usually a couple of chapters on viruses, a couple of chapters on parasites. That is not the name of the game here. My perspective on this is that you can get most of the information you need on any particular agent from Google. What I think we offer, or what we try to offer, is to go beyond that just sort of descriptive cataloging mindset, to try and give you or empower you and give you the knowledge and understanding to really start to appreciate fundamental questions about the science of how infections work, how they spread, what determines outcome, how the immune system works, how we can innovate therapeutically by repurposing the immune system or coming up with new drug targets. We're not going to get that by just giving a sort of endless recitation of lectures on a series of different bacteria or a series of different viruses. What we try to do instead is, as I said, anchor our teaching in current research, where we're asking fundamental questions. And that can be evidenced and illustrated by a wide range of example within infection and immunity, but invariably it's mostly illustrated by examples from the work that's going on here at UCL, because that's what we're doing. So I think that you will, if you do this degree, have a pretty good understanding of how infectious diseases operate, what happens when you get infected, what determines whether you recover or die or something in between. You'll have a pretty good understanding of how the immune system works and what happens when it doesn't work, and how sometimes it can be a driver of disease rather than a controller of disease. But that will be evidenced by a whole sort of different collection of examples that are much more focused on explaining principles than perhaps explaining in detail any individual pathogen. So I hope that answers that question. Mary and Gemma, is that fair?
Yeah.
You can't really disagree, can you?
No.
It's a slightly awkward situation.
I was just going to say that there are some optional modules as well. So well, I mean, for the third year, I can't remember them all from last year, but you can choose more virus focused or more, bacteria focused, and then
That's
obviously
true. That is a
immunology as
good
well.
point.
So
That's
you
a good
can
point.
still tailor your degree if that's what you want to do.
That's a very good point. Thanks, Mary. I'm going to put that slide up actually, because that's quite a useful thing. So to build on that, when you come to year 3, you can choose from this list of modules. Whoops. Oh dear. And that is very much a reflection of what we're doing at the moment. So this one is pretty focused on autoimmunity. That's when the immune system works inappropriately and targets the host, and you get disease. This one is all about how the immune system responds to cancer, and to some extent how we can use the immune system, which doesn't usually respond very well against cancer, and how we can make it respond better. This one, which Gemma will enjoy, is all about using mathematical and computational models to understand biological systems and to use them as a hypothesis generating tool. And that's an increasingly important part of what we do. I said all biology makes sense only through evolution. We have a whole module about evolution and infectious diseases. And if you want to study evolution, it's hard to think of a better context to do that in than infection, because you can watch it in real time. And that graph I showed you of the different SARS-CoV-2 variants is a great example of that. This is about vaccines, how to get rid of viruses. This is really a kind of journal-based module about different bacterial pathogens. This is about how viruses cause disease. And this one is a good one. This is quite new for us. Part of our department is actually an institute in South Africa called the Africa Health Research Institute. And a real strength there and here is work on tuberculosis, which is single, if you set aside COVID, which is a bit of a blip, is now the single most important cause of death through infectious disease in the world. And so that module is jointly delivered between my colleagues here in UCL and some of our colleagues in the institute in South Africa via Zoom. And it's very convenient because they're only an hour different from us. So that can work really well. And that has gone down very well. That really evidences my assertion that we're all about illustrating our teaching with our research. And it's kind of fun to be connecting with the people that are actually delivering this in the research station in South Africa.
Another thing
Okay.
that another
No,
thing
go
that I
ahead,
would
Gemma.
say is that a few of my friends who are from like different courses also end up doing some of ours, and I think some of the year 2 modules were like cell biology, and they were done with like biology BSc or something like that. So there is like a lot of crossover, and like I don't know. I wouldn't worry too much about what you like. I wouldn't worry too much about whether something's going to be covered because I think there's a way to get what you want.
That's right. I mean, clearly, the way we run the program, we know what we've taught you previously, and we know what we expect you to know, and we make sure that you know that. But there is scope, as both Gemma and Mary have said, to steer the degree in a direction that you find interesting, partly by choosing those modules, and partly, and perhaps most importantly, actually, by the project that you do, which makes up a really considerable part of the time you spend in the third year. So how much are you doing each week at the minute, Gemma and Mary, would you say? What's your week look like in sort of breakdown?
Okay, I'm in classes from 9 to 11-ish most days. And then I'm in the lab in the afternoons I'm in class from 9 to 1 most days, sometimes like 9 to 11 or 11 to 1. And then I'm in the lab most afternoons whenever I get out to like 4:30. Or I guess it's only 3 afternoons really. And we've got Friday to just be in the lab, or if you have nothing, you can take a little rest.
Not sure we can condone that. And Mary, are you about the same?
Yeah, it is the same. Yeah.
Okay. And that will go up next term, because next term you have less teaching by design, so you have more time to do your project. Typically what we find is that, and it's slightly dependent on how sort of technically complex the projects are, but some students take essentially the whole of the first term, i.e. now, just to learn how to do the thing, particularly if they're doing a very complex sort of immune staining and processing panel. And then after Christmas, everyone comes back and they've got quite confident and they're quite familiar. And then what they want to do is get into the lab and really crank out the data and find stuff out. And so I think we allow for that by having the taught content of the program shifted so that there's more of it in the first term than there is in the second term. So you can be almost full-time in the lab in the second term, which you will definitely want to do if experience is any guide. Okay. Well, there's only been one question. So this is you've got very short time remaining. If anyone wants to ask questions, now is the time to do it. I'm going to wait 1 minute. Okay. And then, so that's anonymous attendee. I don't know if it's the same anonymous attendee as the previous one. Could you expand on the immunodeficiency and therapies bit? I'm interested in what that part of the course offers. That is a module. Everything is done in modules. So you choose, or you take a certain number of modules and they give you credits, and the credits add up, and that's how you get your degree. The immunodeficiency and therapeutics module is essentially what it says on the tin. It's about how immunodeficiencies work. So immune deficiency is when the immune system is broken in some way. And that can be through infection, and HIV is the classic example. But most of the module is about genetic errors, where you can have a mutation and that breaks one little part of your immune system. And that has very interesting effects in terms of susceptibilities to infection, ability to respond to cancers, and so on and so forth. A big part of the outcome of infection and cancer comes down to your individual immune composition determined by your unique genetic sequences. The therapy part is how to repair that, what we can learn from that to target I suppose simplistically, the broken immune response, either by providing a drug that makes up for it, or possibly by repairing it through sort of genetic type engineering. Right. What would you say is the future of disease spread, or is it too early to tell? That is a very broad question. I mean, I assume you mean infectious disease spread. I think there will always be infectious diseases. There will always be pandemics. There will definitely be another viral pandemic in your lifetime, probably more than one. The more we mess with the planet, as I think Mary said, the greater the likelihood of that happening. I think there is relatively little we can do about it. But what we can do about it scientifically is the kind of research that I described to you in the first part of this talk, where we want to understand how infectious agents interact with their host, what they have to do in order to be able to replicate and transmit. And if we're talking about pandemics, we're talking about viruses, and we're really talking about viruses that come from animals, bats being a good example. So we want to know, what is it that allows one particular virus to jump species and take off and infect 8 billion humans, whereas most viruses do not do that or cannot do that. So I think the future of disease spread is really about studying that kind of thing, and hopefully using the very latest computational approaches, AI, computational structural protein modeling, and so on, in combination to take a really new and properly creative and evidenced approach to that. This is what always happens. We get down to 2 minutes left and everyone starts typing in questions. I'll try and get these two done as quickly as I can. Right, how do exams work? How do they assess you? Well, we use lots of different ways of assessment. Exams are pretty much like exams you've taken. You're in a, well, we've had a bit of a blip for COVID, but now we're back to being in a room with an invigilator. The only difference is that we get you to type the exam on a locked computer rather than write it. But we do all sorts of things. We get you to give talks. We get you to write reviews of papers. We get you to do data analysis. We sometimes get you to do a poster to explain some science. We get you to write a grant application, all sorts of things, all related to the kinds of stuff that we're talking about and doing on a day-to-day basis. Could you please expand on the genetic engineering aspect treating immunodeficiency? Well, yes, I could, but I haven't got time. But very briefly, and this is sort of the Gemma's project touches on this kind of thing. If you have, let's say, I don't know, a defect, a genetic defect that makes your T cells unable to recognize targets, in theory, you can get some cells from that person, repair them in the lab, and then reintroduce those into the patient, perhaps through a stem cell transplant or perhaps just by direct delivery, and that will rescue that deficit. But I would encourage you, if you're interested in that, to Google cell and gene therapy. It's a very exciting and very fast-moving area that is producing really, really exciting, I would even say transformative results in managing these complex diseases. Okay, so we are at the end. I'm just going to, there's one question and I'm going to answer that and then we're going to finish. How did the COVID pandemic impact your department? Well, the whole university closed down, as did the country. And essentially the hospital next door was turned almost completely into a COVID critical care unit. And the virologists in this building and across the road were allowed to work only if they were working on SARS-CoV-2. And so the whole operation was retooled. Our labs were converted so that we could work with respiratory viruses. And everyone worked really, really hard to understand how that virus was operating, how it was able to take off, how it was able to replicate in humans so effectively. And that paper I showed you at the beginning was just one aspect of that. And that work is ongoing as we speak upstairs in this building. And I think it has made a huge contribution to our understanding of how this pandemic has played out and is informing the kind of studies I just talked about in terms of working out what characterizes a pandemic virus and how we might identify the next one. And that's what you want to do with science in the end. You want to have impact and you want to do good. And I think that's a really good example of that. So thank you all for attending. I hope that you found this helpful. Thanks very much to Mary and to Gemma for giving us the student perspective. You can go back to your labs now and carry on with your projects. So thanks everyone, and I am going to finish the meeting now. Thank you all very much and goodbye.